You're listening to Innovating Tomorrow's Medicine with Dr. Jeff Trickett.
In this episode, I'm speaking with David Weitz, founder and CEO of Syrna Therapeutics, the first structure-guided drug discovery company targeting messenger RNA. David is a biotech pharma leader and entrepreneur with over 25 years of experience. Before Syrna, David led Takeda's San Diego research site as part of the research leadership team and also held leadership positions in the legal, IP and BD departments at Takeda. Early in his career, David practiced life science intellectual property law as a partner at Wilson Sonsini. Your early programs focus on immunology and neurodegeneration. What makes these therapeutic areas particularly well suited for messenger RNA targeted approach?
Particularly if you're going to pioneer a new modality, you're going to take on technology risk. So the thing you don't want to do is take on biology risk. So what we've done is we've focused on validated targets where the preclinical and clinical translational playbook is well established.
What's one deeply held belief or assumption about leadership that you've had to completely let go of as you've grown into the role as CEO?
I'm willing to do everything, no matter how menial or how difficult. By showing that you're willing to chip in any and all aspects, you build a great sense of team.
You've been operating in stealth mode. At what point does it make sense to emerge more publicly, and what triggers that decision?
If you feel like you're highly differentiated and you're doing something in a very different way than other folks, it's very useful to stay stealth until you have the data to really demonstrate with credibility what you've been able to accomplish.
Some might call Syrna a platform discovery company, and there are many who say platform companies are out of favor today. How do you think about this, and what's your counter argument to investors?
I think of us as a pipeline company that happens to have a novel modality as a common thread holding our programs together, and then a body of know-how that we've developed that enable us to prosecute it.
So, David, welcome to the show. Great to have you here. Let's dive in with the first question. So congrats firstly on Syrna Therapeutics coming out of stealth mode. In your white paper, you've described covalent translation inhibition of messenger RNA as the next great frontier in small molecule drug discovery. What made you think this was achievable?
Well, first of all, let me say that I started Syrna because I thought that targeting RNA is the next great small molecule target, and that it is just simply inevitable that people will find a path forward, and this will be very important in the future for medicines. Now, people have tried for a very long time to drug RNA, and they've not been as successful as you might hope. Now, what people have largely done is they've been stuck in what I would call target validation. And what they're doing is they're studying how RNA folds and are looking for a site in messenger RNA where a small molecule will produce a desired phenotypic result. Now, the problem is biology associated with messenger RNA on an individual basis is really largely unknown. And they're hunting for a function that they can't really be sure if it's present or it's not present. The other thing is that messenger RNA didn't evolve to accept small molecule ligands. So this small molecule coming along to have a function, on a biomolecule that really wasn't designed to accept it. The other thing is that people have been largely studying how RNA folds in 2D. And so they don't really have a great grasp of what it looks like as a three-dimensional molecule. And there's a lot of speculation about it being multi-conformational, highly flexible. Maybe it adopts different conformations because of RMPs or small molecule binding, or that it changes in cells. Now, we looked at that and said, we really need a new path forward. And so we started with a very simple premise. And that is what we wanted to find was where RNA forms a stable and persistent structured domain, a place that the RNA forms co-transcriptionally and that would stay present, because we felt that something that behaved much more like a discrete biomolecule, like a protein, would be the best place to target with small molecules. Now, we also, Thomas Herman, and my co-founder, and I speculated that the best way to see and identify these structured domains, because people really haven't identified or characterized them in the past, was to use structural biology methods. Because basically, things that were highly entropic were things that would probably not show up. But the things that we could bring into focus with structural biology would be exactly what we were looking for. And we, of course, didn't know how often we would find them. And it turned out that those speculations were correct. And we were able to successfully use structural biology to do that. Now then comes the question of how do you go about drugging them? And that's a new, that's an additional challenge. Now, what we didn't want to do was get caught up in the intricacies of the biology of each messenger RNA. So as a result, we said, what would be a universal mechanism of action that would work for all messenger RNAs? And that led us to covalent translation inhibition. Now let me explain what I mean by covalent translation inhibition. Covalent translation inhibition, we refer to a small molecule that can bind non-covalently to one of the structured domains that we identify. Now we incorporate into that an electrophile which will react with a nucleophile in the binding site to form a covalent adduct. Now once that adduct is formed, when you have a ribosome that comes along and tries to translate that message, it can't pass the adduct. It can't defeat the covalent bond. And as a result, that message isn't able to make protein. And this is true of every single messenger RNA where you can form a covalent adduct. So that's exactly that universal mechanism of action that we wanted to achieve. Now, your question was, why did you think this was achievable? The simple answer is structural biology. Previously, people have been operating without a clear understanding of the sequence of RNA that they were targeting from a traditional three-dimensional structural point of view. But by having structural information that tells us that what we're targeting is stable and persistent, and then by being able to advance programs in a rational, structure-guided manner, we reduce significantly the chemistry hurdles, and we also have significant insights which make prosecution of covalent translation inhibitors in this fashion, much more achievable. And that's what we set out to do. Let me add one other thing, and it's a bit of my history that leads to how I've thought about this. Now, I came to San Diego in 2002. I joined a company called Syrix. They were one of the pioneers in high throughput automation for crystallizing soluble proteins. And they and others, because of that type of innovation, you saw kinase structural biology take off. About 10 years later, people started to solve GPCR structures. And you saw GPCRs, membrane proteins, take off as a class. And so I really felt, and this is what convinced me, that a structural biology solution towards RNA would be exactly the catalyst that would lead to RNA take off as a small molecule drug target.
I think it's very neat, and just at a higher level, we come back to druggable or not, difficult to drug targets, sort of going upstream of proteins, the messenger RNA. As a biochemist originally, I think it's really neat. Let's move on to the next question. So You described Syrna as the first 3D structure-guided drug discovery company for messenger RNA. What makes this approach fundamentally different from how others have targeted RNA with small molecules in the past?
Well, I think you can think of it in terms of form and approach. Now, when you look at most companies that are targeting RNA with small molecules, they fall into a few discrete camps. First of all, you have your splice modifiers. And these companies are following on the heels of risdiplam, where there's an opportunity to interact with an interface between RNA and the spliceosome. And so in that case, they're really targeting that interface. You have people who are targeting RNA with non-covalent small molecules, and as I described, they're hunting to find a function of where the compound would bind and to take advantage of that. And they're interfacing with RNA that, in many instances, they're hoping is flexible so that the small molecule can distort the shape of the RNA into a particular shape to achieve their phenotypic result that they're wanting to achieve. And the third category are degrader companies. And this is the idea that you can bind to RNA and then attract a degrader functionality, typically an RNase, and cause degradation. And in that case, it's interesting because they have the small molecule that binds to the messenger RNA, and then they have a linker that goes out into solvent space, captures this degrading agent, and then hoping that it will circle back, find a different portion of the messenger RNA and degrade it. Now, in our case, our targets don't look like that because we really don't want our targets to be flexible RNA. We don't want we want to have our chemistry, our covalent modification, occur right at the binding site. And we're also targeting RNA that doesn't involve a binding pair like in the case of degraders. Now, recently, we've started to see people adopt covalent mechanisms as their approach. And it's still early days for them. But in their case, they're doing phenotypic screens off of a library of electrophiles. And they're hoping to identify where they can form covalent adducts as a way of identifying targets and then working up into a program. In our case, we're the only ones who pick targets of high promise, find the place to engage with a small molecule in a structure-first manner, and then with rational drug discovery approach, arrive at your covalent inhibitor.
You mentioned identifying druggable structured domains in messenger RNA. What makes a messenger RNA domain druggable, and how do you validate these sites?
Well, the first thing, and I would say the most important thing, is understanding the nature of the target that you are going after. And so again, we use structural biology to confirm that these are stable, persistent, complex structures. And the second thing that we do is we go and we see if we can find hits to them. Now, because we've actually, despite being a very small company, we've done over 20 screens against the same library across different targets. So we have an understanding of compounds that bind to our target that are selective and non-selective. And so we pick the compounds that show that they have selectivity. The third thing is that we build out an SAR around those hits to establish that the binding pocket is complex enough that it can support high affinity. And we've achieved single-digit nanomolar binders, as well as the ability to then accommodate an electrophile, which we can then turn into a covalent translation inhibitor.
So how do your small molecules covalently modify messenger RNA binding sites to block the ribosome? And how do you ensure selectivity for disease-causing proteins?
selectivity is a really important question across all of the approaches to targeting RNA with small molecules. Now, in our case, first of all, we pick structured domains that are actually surprisingly large. Our structured domains are 150 to 300 bases in size. They're stable, they're persistent, and they allow for tight affinity binding. So with that, you get a first degree of selectivity, let's say, because our compounds are binding just to that target. Now, when we look across our lead compounds for the multiple programs that we've addressed, what we see is the Tanimoto score of the lead compounds are quite different. So we see significant chemical diversity in the compounds that bind, which tell us further, confirm the expectation of selectivity. Now, covalent modification is a multiplier of selectivity. We already know from work that other people have done that non-covalent binding generally is silent in having a biological effect. So you need the ability to form that covalent adduct. Now, it's critical that you place the electrophile in the right trajectory and orientation to engage the nucleophile. Now, for off targets, it's highly, highly unlikely that it's going to both bind to your same compound and that the nucleophile will be in just the right place. So as a result, we expect to see an absence of concentrated binding towards a particular off target. Now, let me just point out that one of the things about covalent translation inhibition is that silencing one message eliminates the production of 1,000 or more copies of the protein. So this allows us to actually dose less. And because we don't expect to have reactivity that's concentrated on one specific species, we think through control of dosing and the fact that covalent modification just generally in the cell, it won't be concentrated to give us a specific, safety signal.
So I'm thinking there's a couple of advantages to that approach. Like, firstly, to your earlier point, there's less off-target effects, and then because of the selectivity, you can give less, so it's going to be less toxic. It's better for patients all round.
That's right. And one of the things is I think that the playbook for covalent inhibitors against RNA is different than for proteins. Because in the case of proteins, you're taking advantage of an available nucleophilic base, like a cysteine. The problem is that cysteine is probably present in other family members. However, there's not this same class-based high level of structural homology in the binding pocket when it comes to RNA. So that experience that you have a nucleophile in the same place that you see in the protein environment, we just don't expect to see in the RNA environment.
You've imaged druggable structured domains in the mRNA of greater than 25 disease-causing proteins, far more than anyone else. Can you tell me some interesting things you've learned about them?
Honestly, it's been fascinating to see what they look like. And again, because people really haven't had, genuine 3D structures to the extent that we've had, it really gives us an insight into what we're targeting. So first of all, they're a lot larger than I think people would anticipate, being 150 to 300 bases in size. And the quality of the resolution that we've achieved, shows that they're really quite stable and persistent. Now, the other thing is that we see that there's no sequence homology between the different structured domains. So really, they're quite unique. And it makes sense because it's really not clear what the evolutionary pressure has been to cause them to form. We don't know what their biological function is, or in fact, if they even have a biological function. And the last thing I would point out is that what we see is that these binding pockets form exactly where, from a 2D point of view, you go from a very ordered helical place to this large bulge where you have the bases coming together to form a binding pocket. But relatively speaking, like for example, relative to what you could do with AI in a highly unpredictable manner. And so this is where we're really satisfied by the unique insights that we can get with a structural biology approach that we really don't think anytime soon is going to be available via AI or in silico modeling.
So moving on to targets, modalities, and mechanisms, your early programs focus on immunology and neurodegeneration. What makes these therapeutic areas particularly well suited for messenger RNA targeted approach?
You know, as a biotech, particularly if you're going to pioneer a new modality, you're going to take on technology risk. So the thing you don't want to do is take on biology risk. So what we've done is we've focused on validated targets where the preclinical and clinical translational playbook is well established and is very much well recognized that there are opportunities to shift from, for example, proteins that have only been drugged with biologics to oral molecules and also to molecules that can penetrate the brain. So this has guided us towards a lot of immunology and neuroprotection targets.
How do you think about target selection? Are there classes of proteins like transcription factors or undruggable targets where mRNA inhibition has unique advantages?
There's some really interesting things that you can do on the RNA level that I don't think people have yet had the chance to appreciate. And I really look forward to demonstrating that, with our programs. So let me give you just a couple of examples. So let's say you're thinking about a transmembrane protein, which has an extracellular domain or an intracellular domain. Traditionally, drugs either target the extracellular domain or the intracellular domain. And in the meantime, the signaling for the portion of the protein that you're not targeting is still there. The other thing is a lot of times there's a scaffolding function. So for example, they may form homo or hetero dimers. That scaffolding function is there even if a biologic comes or a small molecule comes and engages a portion of the molecule. Now in our case, what we're doing is we're depleting the presence of that protein. It silences all of the functions that that protein can do. And I think that will yield really unexpected therapeutic advantages. Now, the other idea, which I'm really excited about, is in the area of cancer fusions. Now, cancer fusions, and use BCR-ABL as the most classic one. People target the 3' end of the fusion, which is the expressed protein portion. Now, in our case, we can actually target the 5' pair, and those 5' pairs frequently are involved in very diverse fusions. And so we have the opportunity to be kind of an orthogonal oncology treatment where you're making a drug that can silence the production of proteins associated with whenever that given 5' pair is there. And so these are really fascinating ways that targeting at the RNA level open up doors that previously weren't achievable.
And I guess that opens up a whole range of additional possibilities, targeting the 3', the end, 5' the beginning. So you could do all the combinations, the 3' on its own, the 5' on its own, the 5' and the 3'. So I guess that means you've got a lot more tools in your arsenal to sort of help get better treatments.
Yeah, absolutely. And the other thing is that, let me just also throw out a different aspect, which is significantly, very significant in oncology, and that is the emergence of drug resistance due to mutations. Now, those mutations, just to be clear, happen in the coding region. Now, if you target the non-coding region, you can develop a pan-mutation drug. And you don't have that selection pressure in the coding region when you're targeting the messenger RNA. And this provides another really interesting alternative way to impact therapies.
Yeah, and I guess tying that in with what you were saying earlier, because the non-coding region can have some effect sort of like on the structure and all of that. So it could, if it works, then it could actually make it better for again targeting those regions as well as the coding regions.
Yeah, I mean, I think the thing is that you can have a very complementary approach where on the one hand you're targeting the message, and on the other hand you're targeting the protein product.
I'd like to thank the previous guest for this question. So this is the nice little tradition that we have. So what's one deeply held belief or assumption about leadership that you've had to completely let go of as you've grown into the role as CEO?
I fundamentally think of myself as a team member who picks up all the slack. I'm willing to do everything, no matter how menial or how difficult. By showing that you're willing to chip in any and all aspects, you build a great sense of team.
Yeah, in biotech you're wearing multiple hats anyway, and so that sort of roll up your sleeves mentality really, really important.
I mean, one of the things that has helped me a lot is, I've had a long career before starting Syrna. And I had the opportunity to practice in a law firm. I had the opportunity to be in a startup and sell it to a pharma. I was in a pharma where I was in the legal department, the IP department, the business development department, and even, was a research leader and led the San Diego research site. So I got to sit in all sorts of different seats. And I frequently, for some reason, it's kind of like King Arthur and his court of the round table. And I imagine this very large round table, and each seat is designated for different skill sets. And it's really helpful to have sat in not every, but a lot of those seats. And then when you get to a biotech where you have to wear all those hats, be able to navigate all the different roles that you have to play.
Moving on to platform development and validation, how do you validate that blocking translation of specific mRNA in cells actually translates to the desired therapeutic effect in vivo?
People have this question about why will it work in cells? And what I frequently remind people is that most target validation starts either with genetic evidence, like a loss of function, or with a knockout or an ASO, where you show that reducing protein expression provides the desired phenotypic result. And then what happens is people, let's say, settle and adopt a small molecule or a biologic or some other modality approach to reduce the protein's activity. And so what I would say is what we're doing is really much closer to how you validate targets in the first place. Because with our covalent translation inhibitor, we're going about preventing translation, reducing the amount of that protein, just like you do in the original target validation work or the genetic evidence for the target. And so from that point of view, I think the leap to translation in cells is actually much smaller.
So RNA is commonly characterized as being flexible, multi-conformational, and affected by RNA binding proteins in cells. How do you know your structure domain is present in cells?
We get a 3D view of them. The structured domains that we're focusing on, they form co-transcriptionally in the pre-message. They're present throughout splice modification. They're present in the mature messenger RNA. And when the ribosome, if the ribosome unfolds it and passes, this structured domain refolds. So you'd fully expect that it's present that way. And when we look at the structure by having, solving the 3D structure, what you see is the sequence complexity. If we were talking about proteins, there's just no reason that you would think that a protein would suddenly fly apart and go back to its single chain amino acid state. There's no energetics that would drive that. Take a look at the structure on our website. Why will the helical regions, on either side of where our binding pocket is, why are they going to come apart? There's no energy there for that to occur. And then what we're targeting didn't evolve to accept a small molecule ligand. So there's really no reason to expect a ligand to exist that's going to take that action, or an RNP. And the other thing, let me add to that, is that even if something were to bind to the RNA, it comes on and off. And what we're dealing with here is the energy well. The RNA adopts its preferred state co-transcriptionally, and that's where it wants to be. And anything else is purely a distortion. Now, I like to remind people of how splice modifiers work, when I talk about this subject. Now, in the case of a splice modifier, the splicing event takes fractions of a second as the pre-message is forming. And you have, this small molecule has an incredibly small amount of time to act, because once the splicing event occurs, it's too late. Now, in our case, the target for our small molecule is present in the pre-messenger RNA, is present in the post-translated RNA, it's present, after the ribosome passes. So if you think about the amount of time that's available for our compound to act in comparison to splice modifiers, I think we should be asking splice modifiers how they get their job done so quickly, because I have infinite time in comparison.
What de-risking data do you need to see before nominating a development candidate, and how does that differ from traditional small molecule programs?
So one of the things that we purposely did, and this is why I really like to characterize our efforts as, rational design, structure-guided drug discovery, is de-risking to a very large extent is like any other small molecule program. We're going to subject our compounds to the same ADME, PK, metabolism, distribution, safety studies like everyone else. So from that point of view, it's kind of boring. Now, meanwhile, it's amazing what you can do with protein mass spec and with RNA-seq. So we'll be able to evaluate how our molecule affects protein production with protein mass spec, and we'll be able to monitor, changes in RNA production with RNA-seq. And because we have a structure-guided approach, we're in a great position to tweak our molecule as necessary to engineer out that selectivity. No different than any other small molecule program where you might see some off-target activity that you want to address.
Thinking about oncology, again, that approach for using the RNA-seq would be very powerful because you can compare it to sort of like tumor and then tumor normal like samples. So I think that would be very informative.
Our mechanism of action looks a lot like the way in which you validate targets with either a knockdown or an antisense. We actually have very useful tools to see a signature and an effect on a pathway. And we can use exactly those same those biomarkers moving forward. And for our main programs, which are validated targets, those biomarkers have already been established, and we can very much follow them.
So moving on to business model and strategic discussions, with programs spanning immunology, neurodegeneration, metabolic disease, and oncology, what's your strategy for prioritizing which to advance internally versus partnering?
Well, so one of the powers of our approach, because we don't have a phenotypic screen where we have to live with whatever target comes out the other end, is that we pick targets of high impact to begin with, and then we prosecute them forward. So pretty much all of the targets that we've taken on are targets that we think would be really interesting to develop in our internal pipeline. Now, we've been very fortunate that our approach has been highly efficient, and we've been able to generate chemical series and programs against multiple targets. So we have the position or the ability to be flexible about what we bring in-house, what we keep in-house, and what we partner. But again, I would say what we would prioritize first and foremost would be high-impact validated targets where the translational path is straightforward and which is much easier for a small biotech to do.
So in that sense, you're disease agnostic.
We are disease agnostic, but early on, what we found is that it caused us to lean a lot towards immunology and also CNS indications, a lot of which have an immunology aspect to them.
So you've been operating in stealth mode. At what point does it make sense to emerge more publicly, and what triggers that decision?
We were really thoughtful about and purposeful about being stealth and staying stealth. And I tell people, if you go up into the mountains in Northern California and you were digging gold in the 1850s and you dug out a nugget of gold, I think you'd quietly stick it in your pocket and not attract any attention. You wouldn't go onto social media and start yelling, "dig here." And so I think stealth, especially if you feel like you're highly differentiated and you're doing something in a very different way than other folks, is very useful to stay stealth until you have the data to really demonstrate with credibility what you've been able to accomplish, and where you also have a significant head start. Now, at the same time, there is a point at which you want to reach out to investors and reach out to pharma. And so in our case, make them aware of us and say, hey, we are the most exciting RNA targeting a small molecule company out there. So when you decide who you want to work with, we find ourselves at the top of the list.
Yeah, I think especially for big pharma, the first thing they're going to look at is good science. And then I think I'm always really big on sort of like early engagement. So again, I think having that, this is what we're doing, keeping them sort of like updated. They're actually very happy to hear from you. So I think that makes a lot of sense. Given your background in IP law and your time at Takeda, how are you thinking about building a defensible IP position around this new modality?
I think my answer is going to surprise you. There's the old adage that, a carpenter looks at everything with a saw and someone with a screwdriver thinks everything's a screw. Our strategy is we've filed no patent applications. And we're going to delay filing of applications until we have products to protect when we have compounds that are near candidate selection. Now, what we see is that one of the powerful things is that we've figured out all sorts of aspects of how to do our work. And by just doing more and more of that work and keeping that know-how secret, that's the best barrier that we can establish for what we're doing.
That makes a lot of sense, actually, because at the point where it's really about fast fail. If you've got a winner and then you delay those filings, then it means you have more time for your loss of exclusivity. So I think that makes a lot of sense. What does success look like for Syrna in the next 3 to 5 years? And is it a clinical candidate, partnerships, or proving the modality works broadly?
So my question is, why do I have to choose? I mean, of course, the answer is we want to get to a clinical candidate, get into the clinic. Because first and foremost, we are a pipeline company. I think in the process of doing that, the work that we do is going to demonstrate the effectiveness and all the aspects that we're covering today about why this approach is incredibly powerful. So we satisfied that piece. Now we see that pharma is a really important part of our strategy because we have the benefit of being able to go after more targets than we can prosecute ourselves. And so if we can strike partnerships with people and be able to prosecute more programs in parallel, we accelerate our learning, we become more efficient, we gain further insights, and ultimately we want to be the category leader of what we see covalent translation inhibition as its own modality.
You've talked about potentially kicking off a wave of covalent translation inhibitor companies like Ionis, Moderna, Venus did for their modalities. What does it take to become a modality pioneer versus just another company?
One of the things to recognize is you're paving a road through a forest that's never been cleared. And so there's a lot of things that you have to figure out how to do. And, with that, you have to be really thoughtful, both in interpreting what people have said before and be willing to agree or disagree with the literature. And you also have to be highly, self-critical of the results that you generate. Because you may not have precedental guidance to go with. So you have to embrace a very, innovative environment to go and do that. Now, I'd much prefer, despite it can be hard, I love that innovative leaning aspect of what we're doing. And I'd really much prefer to do that than to be this, one-trick pony company with, one asset that you're betting everything on. Right now you see this enthusiasm in this wave for degrader companies and induced proximity companies. Not that long ago, it was just kind of a concept. And people had lots of questions about, whether it was feasible and how difficult it was going to be and how do you understand, how you design linkers and warheads and deal with selectivity, all sorts of those questions. And now, hats off to companies like Avidity, Chimera, Arvinas, Monte Rosa, who are multi-billion-dollar companies, and that would be a great set of companies to be in, to be with.
Coming from roles at Takeda and Cyrix, which was acquired by Takeda, plus your background as a partner at Wilson Sonsini, how did those experiences shape your approach to building Syrna?
Well, one of the things that I learned from Takeda, which was, I think, really critical, is the importance of time. Really think about how to collapse the time that it takes things. The other thing is that, when you're a service provider in a law firm, you're not quite on the team. And when you get to be in a big pharma company, the team is kind of big. It's easy to lose the mission-driven aspect of it. And so it's a great coming home to work for a small company where you're really passionate about making the difference. You're on a team, you're cultivating your team to work together, partner with really a shared vision and a big goal for what they're trying to accomplish.
So when you build a company from scratch, you get to define its culture. What type of culture are you fostering at Syrna, and how does that differ from what you experienced in Big Pharma?
One of the things that I wanted to instill in the company is the sense of purpose and what a significant scientific achievement and what we can achieve for patients if we can succeed. And it is that purpose-driven environment that we're directly, our small team, is making a difference. I feel that's a critical aspect of what we're doing. The other thing, and it's actually something that I bring from my experience at Takeda. So when I was at Takeda, we had the opportunity to build a 160,000 square foot building. The architects asked me at the time, in order to design the features of the building, they asked me, give me some words to describe the culture. And the one word that I gave them was authenticity. I didn't want gimmicks or cartoonish depictions of what we were trying to be. And I think that authenticity is also important in the culture that we have at Syrna. And it relates to candid conversations that are all meritocracy-driven, science-driven, goal achievement-driven, and just a fundamental genuineness. Plus, as I mentioned, the willingness for everyone in the organization to do whatever it takes to help us succeed. And that's the authenticity that I think is, a really great word to describe what we want to be.
Culture is so important. Having spoken with many CEOs, everything begins and ends with establishing the right culture. So I think that makes a lot of sense. Some might call Syrna a platform discovery company, and there are many who say platform companies are out of favor today. How do you think about this, and what's your counter argument to investors?
I think of us as a pipeline company that happens to have a novel modality as a common thread holding our programs together, and then a body of know-how that we've developed that enable us to prosecute it. So I think that in that sense, I would reject the classical platform definition. Now, what I will say is that every program that we picked had the opportunity to progress because of the quality of the target. So we didn't take on anything just for proof of concept. So we didn't waste our time with that. We always were focused on work on programs that could ultimately turn into candidates that could then reach the clinic and be highly differentiated. And then I think related to that is the work that we have done has been very focused on doing science to address the problems and the uncertainties of what we're facing And so there's very few academic exercises that we've engaged in. And as a result, we've been an incredibly efficient, innovative, pipeline-focused company.
So you can have the best of both then.
One of the things that I think is the problem with the current narrative against pipeline companies is that we don't want just endless repositioning, one-trick pony companies. We see over time that what we think a drug looks like continuously evolves, and there's new drugs, new types of drugs that are emerging, and they don't emerge out of thin air. There need to be companies, and you named a few, and I love those as being my potential future peers. But companies like Ionis, Alnylam, Moderna, the degrader companies, CRISPR. I mean, these are companies that have transformed what you think a drug looks like. And at some level, they had pipeline elements, they had platform elements into them being pipeline drugs, pipeline companies. And I think that is ultimately where huge opportunities in large companies will come from.
What advice would you give to other early stage biotech founders about raising capital in this environment, and what common mistakes do you see?
One of the things that I see, especially in a capital constrained environment, is that people want to claim that they can build what they need to build with less money than they probably need to build it. And I think you have to be very cautious to give in to that pressure and be realistic about how long things are going to take, how much money they're going to take, that you may not get things right the first time, so you need a little buffer, and build that into what your ask is and raise money accordingly. Because if you're underpowered, you're going to only go part of the way, and part of the way is not going to be, it's an unforgiving place to be. So, don't sell yourself short that way. And also think about it in terms of, you want your investors to see you as an effective manager, and an effective manager calls it accurately. They don't skinny things to make it more promising than it is. They make a projection as to time and cost and risks, and then they end up delivering, and hopefully they're spot on or they overachieve. And I just don't see how you can do that if you basically try to raise money by perpetuating a fiction as to cost or time.
You make a lot of good points. I think capital efficiency is obviously very important, but the mistake that I've seen in speaking with CEOs is, to your point, they didn't ask for enough money in the first place. So I think there's always that balance of building some buffer and not asking for too much. But I think especially in the recent years where it's been increasingly difficult to acquire capital, that's definitely one of the mistakes that I've seen made.
Yeah, I think it's one of the mistakes that I keep seeing. And a similar thing is let's say you're a candidate nomination and you want to raise money just enough to do a GLP tox and some CMC. And the thing is, that's not where you need to get in the journey. And you have to be brave enough, as an example, to have the conversation about why what you're building is compelling to get to POC. I would pressure test, is what I'm building something compelling that I should get to POC? And then you can figure out how to stage the money. But just getting enough to answer, to take 2 steps and then think you're going to revisit it, I think is a common mistake.
Can you just clarify what you mean by POC? Is that clinical POC?
Clinical POC.
Yeah, and I think that's a very well trodden path. In terms of getting things to that value inflection point, that's really what you need to do. David, thank you.
Thank you.
Innovating Tomorrow's Medicine with Dr. Jeff Trickett.